Okay, so this is what we discussed the last class. So we discussed about the sources of hits, leads, and candidate drugs. There are three major sources: natural source, synthetic source, and virtual source. So natural sources, again we have so many sources: plant, microorganism. Okay, so for example, synthetic, we have two important methods. One is normal organic traditional synthetic method, and the second method is combinatorial chemistry. So virtual is like using computer for designing a ligand molecule. So do you know what ligand design is an example for virtual world? Okay, so these are the concepts what we have discussed in the previous lecture. So for today's lecture, I'll be talking to you about lead optimization techniques. So before going for the various techniques, let me tell what's the need for lead optimization. So I just want to define the lead optimization again. So it is the process by which a small change in the original molecule is made to improvise the activity. Okay, so this is my own definition, right? So not from textbook. So simply we can say that lead optimization is the process by which we actually make some small change to the structure. So let's take some example. Say, so for example, you have some drug molecule like this. Okay. Say this molecule is having very poor absorption. So how we can improvise the absorption of this molecule by making some small change in the structure of the original molecule. So lack of absorption may be because of poor solubility of the molecule. So what we can do is we can introduce some polar functional group like OH, right? So the addition of this OH, right? So that's a structural difference between molecule A and molecule B. So this small structural change, the addition of this OH, may improvise the solubility of the molecule, so we can expect that more amount of absorption. Okay, so we can say that this process, what I have drawn here, is an example for lead optimization. So again, if you look at the definition, so what we did here is we make very small change in the structure of the molecule. So why do we make this change? Because we need some improvement in the absorption. Okay, so absorption is directly related to the solubility of the molecule. If the molecule is not soluble, it cannot get absorbed, right? So we are trying to improvise the water solubility or lipid solubility. So if you want to make this molecule more lipid soluble, then you can introduce some non-polar functional group, right? So these are examples for lead optimization. Okay, so what's the challenge in lead optimization? Basically, this lead optimization is done with the help of medicinal chemist. Okay, so what's the challenge here? As we know that the drug molecule should have two important property. One is the properties related to pharmacokinetics, and the properties related to pharmacodynamics. Okay, so pharmacokinetics is like absorption, distribution, metabolism, excretion. So drug molecule should have good absorption, distribution, metabolism, excretion. So I hope you studied about details in your medicinal chemistry related to absorption, distribution, metabolism, excretion. Also, the molecule should have good pharmacodynamic property, right? So and the molecule should have very less toxicity. This is what basically we expect for a very good drug-like molecule. Okay, so once if you have a molecule which has good absorption, distribution, metabolism, excretion with very less toxicity and good pharmacodynamic properties, we can say that's a very excellent drug-like molecule, right? So, say for example, you have a molecule, okay, which is having good distribution, good metabolism, good excretion. It's having very less toxic effect. The pharmacodynamic property of the molecule is good. Okay, so everything is fine. The only problem is with, say, so the absorption of this molecule is not good. So what we have to do now? Hope I am connected. Am I
Yes,
audible?
sir.
Yeah.
Yes,
What we
sir.
have to do now? I have other properties are good. Distribution is good. Metabolism is good. Excretion is good. The pharmacodynamic property, the drug action is also good. The molecule is capable of binding and it is able to produce the expected biological response. And good toxicity is very less. The molecule is very good, but only it is having poor absorption. What we have to do now?
So we
Can
can
we
give
say that
it
we should go for lead
intestinally.
optimization? Sorry?
We can give it intravenously.
No, 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 no. We have to. We should go for lead optimization. Okay.
Okay, sir.
We can go for intravenous administration, but if the absorption is not good, again the molecule cannot enter into the cell. Okay, so we have to improvise the if the confusion is related to absorption, say I have good absorption but there is a poor distribution or poor metabolism, what we have to do? We have to go for lead optimization. Do you agree?
Yes, sir.
So why do we go for lead optimization here? We want to improvise the distribution of the drug molecule. Am I right?
Yes, sir.
Okay, so quick response. If you quickly respond, I can go very fast. Okay, so in this example, say we are trying to improvise the distribution of the molecule. So you are making some small change in the structure of the original drug-like molecule. If you do so, you may end up with improvised distribution. But what may happen is that it is not guaranteed that the molecule still will be having same absorption, metabolism, excretion or pharmacodynamic property. Either they can be improvised or no change, or it can be decreased. You would understand. So lead optimization may lead for it may lead for improvised activity. This activity may be anything. We can say absorption, distribution, metabolism, excretion, decrease in toxicity, or drug no action. Anything is activity. So improvised activity or no change. Even after the no change in the structure, no change in the activity, or decreased in activity. Any three can happen, right? The challenging is that even after the lead optimization, right? So the molecule should have very balanced absorption, distribution, metabolism, excretion with less toxicity and good pharmacodynamic properties, right? So that's a kind of a very challenging thing for medicinal chemists. They spend a lot of time on lead optimization to make the molecule having desired properties. That's what I'm trying to say. Okay. So I hope that you have now idea about what is meant by lead optimization. So this lead optimization is not, it's not trial and error method. We have some scientific protocol or procedure or steps. So that is well followed by the pharma company, by the medicinal chemist for optimizing the lead molecule. Okay. So I'm going to share presentation. Is my presentation visible?
Yes, sir.
Yes, sir.
Okay, fine. So Okay, so lead optimization, as I said, that what's the aim? Why do we go for lead optimization? There are two reasons. The one is to optimize the binding interaction with target. Okay, so this is some way related to the pharmacodynamics of the drug molecule. So as you know, pharmacodynamics is so how the drug molecule interacts with the receptor and how it forms the drug receptor complex and how that drug receptor complex lead for the biological response. Okay, so this interaction, right? So if you optimize the interaction, if the interaction is much better, then we can expect much better biological uh, response, drug action. So for that, we can go for the lead optimization. Okay, so the first point is related to the pharmacodynamics, drug action. So if you optimize the binding interaction between the drug molecule and the target receptor, then we can expect increase in the biological response. It's the first thing. The second reason why we can go for is that we can go for increase the activity and reduce the dose level, and to increase the selectivity and reduce the side effects. No need to much worry about this, right? So I'm going to give a lot of examples for you to make it understand, right? So as I said, simply the lead optimization is the process which is mainly done by the medicinal chemist. The main reason for doing lead optimization is improvising the biological activity of the molecule. So here the biological activity, it may be in increasing the absorption, distribution, metabolism, excretion, decreasing the toxicity, or improvising the drug action of the drug molecule. For any of this reason, we can do the lead optimization. So as I said earlier, that this process is not going to be trial and error. So we have some well defined protocol strategies are there. I'm going to show those strategies for you. So what are the commonly used strategies in lead optimization? So these are the list of very commonly used lead optimization strategies. So I'm just going to read one by one. After that, I'll be giving example for each one. Okay, that's what I planned for today's lecture. So the very first thing is varying alkyl substituent. The second strategy is varying aryl substituent. Third strategy is extension. I'll be giving example for all, right? So just I'm reading from the slide. The next method is chain extension or contraction. The next is ring expansion or contraction. Next to that is ring variation. Then isosters, simplification, rigidification. So here we can say isosters or bio isosters. Okay, so these are the very commonly used strategies during the lead optimization techniques to improvising the binding interaction with target. So if we improvise the binding interaction with the target, so we can expect more amount of the biological response. Okay, that's the idea. So let's see, example for each one. What is meant by varying alkyl substituents, right? So let me tell what is meant by alkyl. Okay, so alkyl in organic chemistry, the meaning of alkyl is the compounds which are mainly made up of they'll be having only carbon and hydrogen. Okay, those are called as alkyl groups. So if I say CH3, it's called as a methyl group, and this is an example for alkyl group because it is made up of only carbon and hydrogen. Okay, that is the example for alkyl group, right? So now if you take any drug molecule, they're basically organic in nature. Yeah. Just a minute, okay. Hello.
Yes, sir.
Yeah, sorry. I got a call. Okay. So is my screen visible?
Yes, sir. It is.
Yeah. Okay. Yeah. Um, no. So first is the alkyl substituent. Okay. Alkyl groups are they're mainly made up of carbon and hydrogen, right? So example is CH3. Okay. So this is methyl group. Okay. So see here, this region is uh, okay. So this region is the receptor, and this white color region is this is the binding site, and you have the lead compound here. So the lead compound is having side chain. What type of side chain is this? Can we say this as alkyl substituent?
Hydrophobic. Sir.
Am I right? So if you remember the previous lecture or before to that, right? So we discussed about various types of molecular interaction. Do you remember?
Yes, sir.
So what is the nature of this binding site? If you take globular protein, okay. So the nature of this globular protein is that the interior part of the globular protein will be hydrophobic in nature, and the exterior part will be hydrophilic in nature. Is this region interior part or exterior part of the protein? I am taking very simple examples, expecting some answer. Is it hydrophobic region or hydrophilic region? The binding site.
Hydrophobic, sir.
Yeah, the interior part is hydrophobic in nature. Okay. So the interior part is hydrophobic in nature. So this methyl group, so what type of interaction is possible between this binding site and this methyl group? Can we say hydrophobic interaction is possible?
Yes, sir.
Yes, sir.
Yes, right. So here, how many methyl groups we have? We have only one methyl group. So the number of interaction, the intensity of the interaction will be very less. Okay. So here, if you take the same lead compound, we can say this is analog of the lead compound. Okay. So instead of one methyl group, now this is replaced by 3 methyl groups. Okay. Still, there is a space for fitting all the 3 methyl groups. It can the binding pocket can accompany all the 3 methyl groups. So what will be the intensity of the interaction now? Will it be increased or decreased?
Increase, sir.
It is now increased, right? So the intensity of the hydrophobic interaction is now increased, right? So if the intensity, if the interaction is very strong, we can say this is stronger comparing to this one. If it is stronger, so in which case we can expect more amount of biological response. The first case or second
Second
case?
one,
Second
sir.
one, right? So second one will be having more, we can expect, right? So we cannot guarantee, we can expect more amount of biological response, because this time the interaction between the drug molecule, drug like molecule, and the binding site is very, very intense. Or the fit between the drug like molecule and the binding site is, it's a very tight fit comparing to the first example. And this type of optimization is what is called as varying alkyl substituent. Is it understandable?
Yes, sir.
So
Yes,
what is the
sir.
basic requirement for going for varying alkyl substituent? So now what do you think is the basic requirement for going for alkyl substituent? Or do you agree that we need some
More
space in
binding
the binding
interaction.
site? Basic criteria is that we need some space in the binding site, right? For
Yes, sir.
for fixing more number of alkyl groups. That's a basic requirement for going with varying alkyl substituents. Okay. So the next is varying. Okay, this is another example. I just want to skip this, right? So okay. Um, okay, we'll have this example. So second is also example for varying alkyl substituent, but this time, sometimes you may want to improvise the selectivity of your drug-like molecule. Okay, so for improvising the selectivity of the drug-like molecule, we can go for varying the length of alkyl group, length, also the bulkiness of the alkyl group. So I'm going to show one example for you, right? So here we have two receptors which are labeled as receptor 1 and receptor 2. Receptor 1, and this is receptor 2. Okay. So do you find any difference in the architecture of the receptor 1 and receptor 2? Or do you find any difference in the architecture
Yes, sir.
of the binding site of receptor 1 and receptor 2? What differences you find?
Binding region is varying.
Height, sir.
What do you, which region do you say as the binding region?
That
The yellow color, right?
yes, sir.
Okay, so absolutely right. The binding region in receptor 1 and receptor 2, they are not same, they are different. Okay, so here you can see the binding region in receptor 1 is present here, and the binding region in the receptor 2 is present here. That's a slight variation in the position of the binding region present in the receptor. That's a point. So now what we are going to do is that we are going to fix a molecule inside the binding region. Okay, so say we have two molecules now. I'm going to show the first molecule and second molecule. So let's number this for better discussion. So let's say we have, um, so let's say this as the first molecule. Let's say this is the first molecule and this is the second molecule. Okay. So which of this molecule can be fitted in receptor 1? So the condition here is that the nitrogen of the drug molecule should go and bind to the binding region, because that's a corresponding binding region for N. If you do so, can we fit both the molecules in this binding site or only one molecule?
We can fit both, sir, but first will be more interactive, I guess.
First will be more interactive or second will be more interactive? Which one is?
First one, sir, because it occupies the full empty space there.
Okay, so I okay, fine, that's good. That's a good guess. Okay, so do you people agree that both we can fit? Yes, both
Yes,
we
sir.
can fit. Can we do the same thing for if we try to do the same thing for receptor 2? What will happen? Can we fit both the molecules in receptor 2 or not? Can we fit or not?
Only two.
Only 2 can be fitted in
Only
receptor 2. We cannot fit 1.
Yes.
What's the reason? How do you say that the first molecule cannot be fitted in receptor 2?
Space is not.
Space is not enough. In medicinal chemistry, we call it as there will be a type of steric clash. Okay, the space here is not enough for accompanying this first molecule, but still we can fit the second molecule. So N comes here. So you have two groups here, and CH3 goes here. But in the same way, we cannot fit the first molecule. That's a story, right? So now coming to the structure of first molecule and second molecule, what is the structural difference between first molecule and second molecule?
Where
we
alkyl
say-
substituent.
Yeah, can we say first molecule is kind of variation in the alkyl
Length,
substituent?
sir.
Am I right
Yes.
or wrong?
Yes, sir.
So one additional
Yes, sir.
CH2 group is here, right? So varying alkyl substituent. So what's the point is that varying the alkyl substituents will lead for selectivity. Now my question is, compound 1 and compound 2, which of this molecule is not selective? Which of this molecule is not selective for receptor 1 or receptor 2? One or two? Which of this is not selective, which can go and bind to both the receptor?
Second one, sir.
Second one, sir.
Which is selective? First one is first molecule is selective. It can go and bind only to the first receptor, not to the second receptor. You would understand?
Yes, sir.
Yeah, that's so. The point what I'm emphasizing here is that sometimes varying the alkyl substituents can be used for introducing the selectivity. Okay, we can improvise the selectivity of the molecule using varying alkyl substituents. So, thing, right? So yeah, this is what I said. So here we have a steric block. That's why we cannot fit the first molecule inside the binding site. Okay. Right. So moving to a real-time example. So we have adrenaline, and we have salbutamol, and we have propanolol. Among these three compounds, two compounds already we studied. Do you remember which two compounds? In one of my lecture, I discussed about two of the compounds which are mentioned in this slide. Which two compounds?
Adrenaline, salbutamol.
Adrenaline and salbutamol, right? So now I'm showing the structure of adrenaline and salbutamol. Can you tell what is the structural difference between adrenaline and salbutamol? In two places, you can see the difference. Can you locate where the difference is? Can we say here we have the difference between adrenaline and salbutamol? Okay, so can we say here we have some difference between adrenaline and salbutamol?
Yes, sir.
Yes, right? Any other difference anywhere do you find between the structure of adrenaline and salbutamol? You cannot find. Am I right?
No, sir.
Okay. So now, what is the difference between these two groups? Can we say this as varying alkyl substituent?
Yes, sir.
Yeah, this is an example for varying alkyl substituents, right? So I have some interesting slide for you, right? So to make you to understand. So this receptor is called as alpha adrenoceptor, or sometimes called as alpha receptor. Okay. So two types of receptors there. One is alpha receptor, alpha adrenoreceptor. Another one is beta receptors or beta adrenoreceptor. So here what is illustrated is the architecture of the binding site of the alpha adrenoreceptor, and you can see some color encircled, right? So those regions are the binding regions which are present in the target site or within the receptor. Okay, so I just want to show the labeling of each binding region, characteristics of each binding region. So this violet color is the ionic binding region, the orange color is hydrogen bonding region, this orange is hydrogen bonding, one more orange color is also hydrogen bonding, green color is van der Waals bonding region. Okay, so here we have a small molecule, adrenaline. Okay, so are you able to see can we say adrenaline is well fitted inside alpha receptor?
Yes, sir.
Can we say it is having shape complementary with the binding site architecture of alpha receptor? Yes or no?
Yes, sir.
Yes, sir.
Okay, is it having a binding complementary? Because in the previous slide I have shown the various binding regions in the receptor. So can you check for the binding complementary? So can we say what is this region? This region. What is this region? This region
Hydrogen.
corresponding for hydrogen bonding. What is this atom?
Oxygen, I
right?
guess.
Can it make interaction or not? If you have
Yes, sir.
hydrogen bond donor here, can it make interaction or not?
Yes, sir.
Yes, it can make. So we can say here we have the binding complementary. Can we say here also we have binding complementary?
Yes, sir.
Yes, sir.
So can we say here also we have binding complementary?
Yes, sir.
Yes. So what is this violet color region? This corresponding for
Ionic interactions.
ionic interaction. So here can we say nitrogen atom is strong electronegative and there is possibility for interaction between these two groups? Can we say ionic interaction is possible here?
Yes, sir.
Yes, right. So what is the green color region?
Wonder.
Huh?
Wonder Waals, sir.
Van der Waals interaction. So what is the nature of this phenyl ring? It can participate in the Van der Waals interaction or hydrophobic interaction. Am I right? So we can say that the molecule adrenaline is having very good fit with alpha adrenoreceptor. Also, it is having all the necessary binding complementarity, right? So it's a very good molecule fitted well inside the binding site of alpha receptor. So you have to keep this image in mind, right? So I'm going to show the architecture of beta receptor, and you have to tell what's the difference between alpha adrenoreceptor and beta adrenoreceptor. Okay, so do you find any difference between the alpha receptor and beta receptor? Again, I show this is alpha receptor, and the one that I show in this image is beta receptor. Do you find any difference in the architecture of the binding site of the protein molecule?
Yes, sir.
So what is the difference? So can we say we have some difference in this region?
Yes, sir.
So can we say that this beta receptor is having some additional binding volume comparing to alpha receptor? Again, I repeat, can we say that the beta receptor is having some additional binding volume, binding site volume?
Yes, sir. It is having more volume.
Yes, sir.
So do you find any difference in the binding region of alpha receptor and beta receptor?
No, sir.
Do you find any difference in the binding regions of alpha receptor and beta receptor? Are they same or different?
Same.
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 They are same. If they are same, the question now is, can I fit adrenaline in this beta receptor or not?
We can fit it, sir.
Okay, we can fit it, right? So that's what I'm going to show to you. So again, I am bringing the adrenaline molecule. Here you can see how well it is fitted. Is it having shape complementary? Is it having shape complementary?
Yes, sir.
Is it having binding complementaries like alpha receptor?
Yes, sir.
Yes, sir.
So can we say that adrenaline is not selective for alpha or beta? It can bind to both alpha and beta receptor.
Yes, sir.
Okay, very good. So if you want to make the molecule very selective for beta receptor, what we have to do? Say we need a molecule which is very selective for beta. It should not go and bind to alpha. What we have to do?
So making it
Can we take adrenaline as the starting material?
yes, sir. We can make.
Can we do lead optimization on adrenaline?
Yes, sir.
What type of lead optimization we have to do?
So at the right end, it is having some space to be filled. No, sir. We can add some molecules so that it fills that gap.
What using what we can fill? That's a question. Can we add? So, do you agree that this additional space is hydrophobic in nature?
Yes, sir.
Can we go for varying alkyl substituents?
Yes, sir. We can.
Okay, replacing one CH3 with more number of CH3, right? So, do you agree that there is a variation in this region?
Yes, sir.
So, what is this structure? This structure is no more adrenaline. This is called as salbutamol. Am I right?
Yes, sir.
So,
Yes,
can
sir.
we fit this salbutamol in alpha adrenoceptor?
No, sir.
No,
No. Okay, so this salbutamol is very selective for beta adrenoceptor. We cannot fit this molecule inside the alpha adrenoceptor. So again, the concept is like I just want to repeat the same. So in case of adrenaline, so this molecule can go and bind to both alpha receptor and beta receptor. So we can say lack of selectivity. This is called as lack of selectivity. But if you take salbutamol, so this molecule can specifically go and bind to the beta receptor, not to the alpha. So it's very selective or specific for the beta receptor, not to the alpha. So selectivity of the molecule is achieved with the help of varying alkyl substituents. You would understand?
Yes, sir.
Okay, so one more illustration I want to show, right? So now this is alpha receptor. We are trying to fit salbutamol in alpha receptor. Definitely we cannot fit. You can see some kind of clash here. Okay, are you able to see this? Is it understandable? There's a kind of clash here.
yes,
Okay,
sir.
so that's the point, right? So that is alkyl varying alkyl substituent, right? So I don't want to go to the chemistry part, right? So just I'm skipping those. So the next concept is called as varying aryl substituent. Let me tell what is meant by aryl substituent first. Yeah. So aryl means anybody know what is meant by aryl substitution?
Ring structures, sir.
Huh?
Ring
What is aryl?
structures.
Okay, absolutely right. So it's kind of a ring structure. So aryl means that the molecule will be having ring on it, right? So say for example, what is this molecule? Can we say this is benzene? Can we say this is chlorobenzene?
Yes, sir.
Say for example, if you have OH here, right? So I can keep this Cl either in this position or this position or this position or this position. So as you studied in your basic organic chemistry, we have with respect to this OH we have alpha position. So these two positions are alpha position. These two positions are beta position. Sorry, this is ortho. This is meta position, and this is para position, right? So which means that we can change the position of the substituent with respect to some functional group. That is what is called as varying aryl substituents, right? So here I have an example. So we have a phenyl ring to which OH group is attached, and substitution at para position is there. Okay, there is a substitution at para position. Okay, So this is ortho position, and this is meta position, and this is the para position, right? Uh, okay. So with respect to this, y, this is ortho position, this is meta position, and this is para position. In both way, we can write. Okay. So here we have OH, and here we have binding site. We have some region which has a hydrogen bond acceptor. So what type of interaction is possible between these two groups? So there is possibility for hydrogen bonding. Okay. But in this case, the strength of the hydrogen bonding will be kind of weak hydrogen bonding, because the strength of the hydrogen bonding depends on the distance and the angle. The angle is not suitable for the formation of hydrogen bonding, so it could be a weak hydrogen bonding. So in the second case, right? So what I'm interested is, can you tell what is the structural difference between the first molecule and second molecule? If I say this is first molecule and this is second molecule, what is the structural difference?
Position of OH, sir.
The position of OH now in which position? With respect to this Y, it is now in
Meta position.
meta position, right? So if I change the OH from para to meta position, so now can I say that the interaction will be stronger in this case? Because it is having optimal distance and angle for the formation of hydrogen bonding. Can we expect stronger interaction in case of second structure comparing to the first structure?
Yes, sir.
Yes,
Yes,
right?
sir.
So what we did is that we use the concept called as varying aryl substituents. Just now I told what is meant by aryl substituents, right? So varying the aryl substituent position of the functional group will lead for increasing in the strength of the binding. So ultimately, if there is an increase in the strength of binding between the drug molecule and drug target, what we can expect? What will happen for the biological activity? So can we expect increase in the biological activity?
Yes,
Okay,
sir.
and then
Yes, right. Any question? Are you able to understand?
Yes, sir.
Okay, fine.
Yes, sir.
Any question? I think somebody asked something. Okay, I assume no questions, right? So this is called as varying aryl substituent, right? So moving on to the next example. Next is called as extension. The meaning of extension is, adding extra functional group. The meaning of extension is adding some extra functional group. Various functional groups like, what's the name of this functional group?
Hydroxyl.
Hydroxyl group. What is the name of this functional group?
Carboxylic acid.
Carboxylic acid, right? So we have so many number of important functional groups in organic chemistry, right? So the concept what we are going to use here for the lead optimization is called as extension. Literal meaning of extension is adding some extra functional group, right? So I'm going to give some example for you, right? So everything, I simplified using some graphical illustration, right? So assume that you have receptor binding site like this. So this yellow color region and this yellow color and this yellow color regions are the corresponding binding regions which are present in the receptor. Okay, so the drug molecule should come and make interaction with this region, and this region. So let us assume that we have a drug-like molecule like this. So where you have three functional groups which are oriented towards the three yellow color region, binding region of the drug target. So can we expect interaction between all the three groups? Yes or no?
Yes, sir.
We can expect the interaction, right? The question is, can we improvise this interaction? So I'm going to show the modified version of this receptor binding site. Let us say that you have one more binding site here. Now, what I have to do if I want to improvise the strength of the interaction? What we have to do if we want to improvise the strength of the interaction between the drug molecule and the binding site?
We should use that unused binding region also, sir.
Absolutely right. Who answered this?
Vasavi, sir.
Okay, it is better you can tell your name, right? So again, I don't know who is actually answering. I am getting much better answers, right? So this should be, this is not well used by the drug molecule. We can use this space well So what we have to do is take this drug molecule and go for the lead optimization. What type of optimization we can go for? We cannot go for the simple, varying the alkyl substituents. In this example, we have a specific binding region. So what we can do is, we can make some change in the drug molecule, such a way that we can introduce some other functional group in this position that can interact with the unused binding region. So that is what is illustrated here. See here. So now I'm going to label this as compound A and this is compound B. So before the lead optimization and after the lead optimization. So which molecule will have stronger interaction with the binding site?
B, sir.
B, right? So for which analog we can expect more biological activity?
B.
B, right? So how we
B, sir.
achieved the improvisation in the biological activity using the lead optimization technique called as extension or adding extra functional group. This is an example for, I just want to give one real-time example, right? You can understand how well this concept is used in real drug discovery process. So this ACE inhibitor, ACE is an enzyme called as angiotensin converting enzyme, one of the very important drug target to control the blood pressure, right? Um, do you remember I said one drug molecule name which act as inhibitor for ACE? When I talk about various sources of hits, leads, and candidate drug, one of the slide I specifically talked about ACE inhibitor. Do you remember what's the name of the drug molecule? So if you people have the habit of writing lecture note, you can refer and tell. What's the name of the drug molecule I discussed as inhibitor of ACE, angiotensin converting enzyme?
Captopril, sir.
Absolutely right. So it's called as captopril. Okay, so that's a drug molecule which is very standard drug molecule that inhibits angiotensin converting enzyme for hypertension, right? So okay, so this is the graphical abstract or illustration of the drug molecule inside the binding site of ACE enzyme. Okay, this enzyme is angiotensin converting enzyme, and this is a drug molecule, small drug-like molecule inside. I'm going to show the binding architecture of the enzyme. Okay, so the binding architecture is like here. There is a huge vacant space which is not well used by the drug molecule, right? So if you want to improvise the strength of the interaction between drug molecule and the hydrophobic pocket, what we have to do? What we have to do? Do you agree that we have to make use of this space?
Yes, sir.
Yes, right. So we can go for either varying the alkyl substitution, but how much alkyl groups you're going to use. It's a big question for this example, for this illustration, right? So better what we can do is we can go for adding some extra functional group. So just have a look on this. Can we say this is example for extension?
Yes, sir.
So what are the groups which are added extra? So here you have CH2 group, here you have CH2 group, and here you have C6H5, a phenyl ring. So which one we can expect to have very stronger interaction?
Second one,
one,
sir.
Second one, right? So in this case also, the second one we can expect the molecule will be having stronger interaction with the binding pocket of angiotensin converting enzyme comparing to the first one, because the binding pocket, the hydrophobic region is well utilized or used by the second compound comparing to the first compound. This is not just illustration, right? So it's a real example. Okay, this is an approved drug molecule. This is the way how they optimize the ligand molecule to improvise the activity of this molecule. Okay, so this concept is what is called as extension or adding extra functional group.
So what is the name of the functional group added, sir?
We cannot name. We can name functional group in a different way. Anyway, okay, I'll give answer for you. Okay. Yeah. So the extra functional group added here is okay. This is extra functional group added. CH2, okay, CH2 and C6H5. So I can rewrite this in a different way. CH2, CH2. This I can draw as a cyclic ring structure. Both are same, right? So we can say, can you tell what name we can give for this substitution?
Ethyl, sir.
Huh?
Ethyl.
This is okay. Along with this phenyl, C6H5, CH2, CH2. Hmm?
Ethyl.
Actually, not audible. Can you people speak a little louder? I hope my voice is audible for you. Can we say this as phenyl derivative
Yes, sir.
or alkyl derivative or ethyl derivative? Anyway, right? So we can give many names for this functional group, right? Even if you split this into small partitions, right? So we can say this as a phenyl functional group or benzene group is there. This too we can call it as alkyl groups, right? Many ways we can say, right? Because there is no single functional group here, right? So it's a kind of small scaffold is introduced into the structure. That's what I can say. So this is another example, real-time example, in which they went for adding some extra functional group to optimize the molecule. Serine into ecothiopate is potential molecule, right? So what is the extra functional group they have added? So we can say this region is completely extra. They have added to the original drug-like molecule or lead molecule. So we can say this is a kind of lead optimization in which they have used adding extra functional groups. Understandable?
Yes, sir.
If any question I can address, I think this is enough for today's class. We have 5-6 more methods I'll be discussing in the next class. So any question up to this I can answer. Any question related to today's session or previous session or any general question I can answer. I think we have 1 or 2 more minutes. Any questions? Okay, if no question, okay, fine. If no question, you just go through the material, right? So if any question, we can discuss in the next class. Okay, so I think we can wind up this session. Thank you.
Thank you, sir.
Yeah.
Thank you, sir.